Welcome, very welcome to everyone joining us today at our Cochrane webinar on synthesizing evidence from human genetic association studies. We are delighted to have Dr. Kimona Rampersadh with us as our presenter. So Dr. Kimona Rampersadh is a research fellow at the Cochrane South African Centre, where she is focusing on synthesizing evidence on genetically associated diseases to inform the public and public policy as well as practice. She holds a PhD in the Faculty of Medicine from the University of Cape Town and has a background in medical microbiology. Her research spans antimicrobial resistant Streptococcus A rheumatic heart disease while employing both laboratory and epidemiological approaches. Dr. Rampersadh has published systematic reviews and meta-analysis on antibiotic susceptibility and virulence factors. And she has also presented the findings at national and international conferences. Before we begin, we'd like to bring a few pointers to your attention. Please note that this session will be recorded for learning and dissemination purposes. However, the recording will be used responsibly and in line with the SAMRC privacy policy and POPIA guidelines. So by remaining in the session, you acknowledge and consent to the recording. To ensure that the webinar runs smoothly, colleagues, here's a few house rules. Please keep your microphones muted unless you are invited to speak and to avoid background noise. Please make sure to use the chat box if you want to share comments or post questions at any time. Kimona will address them after talk. And if you'd like to ask a question verbally, please raise your hand first and then you'll be called on to speak. Also, we encourage respectful engagement and professional courtesy to create a positive and inclusive learning environment for everyone. And lastly, we ask you to stay connected until the end as we have some important discussions happening as well as resources to share. As a final point, enjoy the webinar and I trust it will be insightful. On to Kimona.
Thanks very much, Benny. Um, so can I start with the presentation so long? Are there enough attendees?
Yes, we may start. We have 33 online.
Okay, thank you. So good afternoon, everyone, and welcome to this webinar. I'm so happy to present to you today. So we all know that there's been an increase in the number of publications on human gene association studies, and therefore there is a need for evidence synthesis reviews. In this webinar, we will be looking at synthesizing evidence from human genomic association studies, and this will help you make informed decisions when conducting your own gene association reviews. So here are the key objectives for today's session. First, I'll explain what HuGE reviews are, so their definition. We'll talk about why they're important, why individual genetic studies aren't enough. Then I'll give you an overview of the HuGEnet review handbook, and I'll take you through conducting a HuGEnet review. And finally, we'll look at some exemplars of published reviews that illustrate the process in practice. So what is a HuGE review? So let's start with the basics. If you could please scan the barcode and vote in the poll of what you think the answer could be of what a HuGE review is. And yeah, so is it A, a systematic review of genetic associations and their impact on health? Is it B, a clinical guideline for prescribing gene-based therapies in humans? C, an evidence-based summary of gene disease links based on population studies? D, it details human uniqueness in genetic expression Or is it E, both A and C? So I'll give you 2 minutes to do so. So if you could please again scan the barcode and vote on the poll of what you think the answer could be.
Excuse me, Kimona. Some people are saying that the barcode is not working. So would it be possible if they post it in the chat?
Yes, that's possible. Yes, please do
Okay.
so.
Colleagues, if the barcode is not working, you can post the answer in the chat. We will monitor it. Thank you.
Benny, if you could please let me know what the responses are, that would be great.
I will do so.
Thanks.
Okay, Kimona, most of the people chose E. Kim, Sarah, Emma, and Mastima.
Okay, perfect. So I think I'll go on to the next slide. And that is correct. It is indeed A and C. So A, a systematic review of genetic associations and their impact on health is indeed true. It's a synthesis of genetic data across populations to inform disease risk, prevention, and health policy. B is in fact false, and this is because it is a summary of genetic associations to inform public health, not to direct treatment decisions or prescribe therapies. C is indeed true. Huge Review compiles evidence from population-based studies to summarize how specific genes are linked to diseases, helping inform public health strategies. And D is in fact false. Huge Review stands for Human Genetic Epidemiology Review, and it's not human uniqueness in genetic expression. So why are huge reviews important? So the purpose of a huge review is to integrate population-based genetic epidemiology studies to understand genetic influences on health and disease. Similar to traditional systematic reviews, you would apply systematic and transparent methods and use meta-analysis. In this case, it would be disease associations to identify genetic contributions to complex diseases, with the overarching goal of synthesizing knowledge for evidence-based medicine and public health. Why are huge reviews important? Firstly, they standardize review methodology by ensuring consistency and transparency. They improve evidence reliability through statistical meta-analyses and critical appraisal tools. They facilitate reproducibility by enabling independent verification through structured synthesis. And lastly, They provide actionable knowledge for stakeholders by translating data into insights for clinical and public health decision-making. To make this possible, we need structure, and that's where the HuGENet Review Handbook comes in. So this was the first formal organization of methods for undertaking systematic reviews of gene association studies. I won't go into every detail here, but keep in mind that this handbook is the go-to resource for anybody wanting to conduct a huge review. The handbook covers everything from how to frame research questions to how to search, extract data, assess bias, conduct meta-analyses, as well as interpretation. The handbook details the general format of huge association reviews, guiding you through each step in the review. The abstract details the gene and the associated disease or disorder, and the background or introduction of a huge review should include details like the genes, the genetic variants and their frequency, as well as the diseases or other diseases or outcomes. It also goes on to explain the type of objectives you would include and the methods, such as selection criteria, identifying the studies, and the data extraction form. So what type of data you'd be extracting for a huge review. It details the results, so the layout, traditional layout of the results when writing a huge review. So the first paragraph would be the included studies, and the second paragraph would be the quality and methodology of studies, and so on. It also details the discussion section. So the summary of the findings, what type of findings you would reveal, and the limitations of the huge review. It also mentions the biology. So what is the mechanistic evidence that's relevant to the association, and the potential public health impact and other implications of the results. So a huge review follows the same process of a traditional systematic review and also uses the PRISMA guidelines. For those of you who don't know what PRISMA stands for, PRISMA is the acronym for Preferred Reporting Items for Systematic Reviews and Meta-Analyses. So the definition is very much similar. So the huge review definition is very much similar to a systematic review definition. It differs in the fact that it's an evidence-based assessment of genetic association studies that synthesizes data on how human genetic variants influence health and disease. The aim of the review is to summarize gene-disease associations from individual studies and their implications for health, for disease prevention and population health. The unit of analysis is very much similar to traditional systematic reviews, where it includes primary studies, but in this case, it would be population-based genetic publications. And the type of analysis is also a meta-analysis, but this would be specific to the disease state and genetic associations or allele frequency, or in the case of environmental interactions, the gene-environmental interactions. So there are clear components that you would include in the huge review. And firstly, it would state the review question. So this would be defining the genetic association being investigated and its relevance to public health. This can also be a pharmacological phenomenon. So you would go on to explain that in your inclusion criteria. So for the inclusion criteria, these are population-based genetic studies that are eligible. In this case, it would be case control studies that you would mainly include. And this type of search strategy that you would do or conduct would describe systematic methods for identifying relevant genetic epidemiology literature. And you try and use an extensive search strategy, which uses multiple databases. To screen the articles, you would ensure that it meets the inclusion criteria, and you would concentrate on the elements specific to the PICO elements, which I'll explain later on in the presentation. For the data extraction, the type of data you would extract for a HuGE review would be key variables, such as the allele frequencies, the effect size, the study population characteristics, the methodological approach. This would be the study design and the sampling method, the methodological quality of the included studies. So what risk of bias tool you would include. In some cases, it can be the Newcastle-Ottawa scale tool, and then also the study design. And so the risk of bias tool would depend on the study design. And then lastly, you would record the effects of the intervention with the results presented by outcome, so the allele frequencies and the odds ratios. I just would also like to mention that when you are conducting a HuGE review, you would also need to register the protocol through Prospero and ideally the HuGEnet database. So when phrasing the questions, you would use the mnemonic PECO. So P in this case would be the population or the patient or person, E is the exposure, C is the comparator, and O is the outcome. So for example, in a population of looking at humans with the exposure of vitamin D polymorphism and the outcome of a TB susceptibility, you would ask the question, are people carrying vitamin D receptor gene polymorphisms more susceptible to tuberculosis? So there are different types of review questions, but with a HuGE review, the main type of question you would ask yourself is a risk-associated question. So the typical question would be, what factors increase the risk of this disease? So there are different types of HuGE review questions. Firstly, there's a genetic association question, and this focuses very much on whether a genetic variant is associated with a disease or a trait. So if you break it up into the PECO elements, where population is adults of European ancestry, the exposure is the presence of the APOE epsilon 4 allele, and the comparator is the absence of that. The outcome is the risk of developing Alzheimer's disease. And using this PECO elements or the PECO mnemonic, you would formulate your review question. And in this case, the question would read, in adults of European ancestry, is the APOE epsilon 4 allele associated with increased risk of Alzheimer's disease? So I'm not sure whether the barcode would work in this case, but if you could, I'm going to read the scenario for this next type of HuGE review question, which is gene-environment interactions. So the scenario reads, a colleague mentioned reading that lung cancer risk in smokers aged 40 to 65 years old may be related to modification of the GSTM1 null genotype. And you decide to do a HuGE review to investigate how genetic variants interact with environmental exposures to influence outcomes. So based on the scenario, can you determine the PECO elements? If you could please go to the chat and put in these elements of what you think the PECO is in the scenario. So again, P would be your population, E would be your exposure, and C would be your comparator, and lastly, O would be your outcome of interest. So Benny, if you could please give me an idea of what the comments are in the chat.
I will do so. At this point, no one has entered anything in the chat. They're probably still thinking about the answers.
No
Right,
worries.
colleagues?
We'll give them 2 minutes to do so.
But you can keep an eye on Menti as well. Maybe some of them entered it there.
Mm.
Um, we have two very exciting participants. Masima said to be smokers aged 42 foot 65. And then Sarah Cooper said that the population is smokers aged 40 to 65.
Sounds good. Any idea of the exposure? This is a gene environment interaction, so that should give you an idea.
Kimona Kim said GSTM1 modification would be the exposure, if I'm not mistaken.
Yeah.
And then we have Omar, Dr. Omar, who said the P smokers is GSTME, C is the environment, and lung cancer.
Outcome is the outcome lung cancer.
Yeah.
Okay.
And then we have Mark Engel, who said EXP GSTM modification. The control is no GSTM1 modification. Kim said C, which is the comparator non-GSTM1 modification. Great minds think alike. And then Kim said O would be lung cancer.
Okay. So shall I move on? I can explain the answer. Sounds good. So P is indeed smokers aged 40 to 65. E would be carriers of the GSTM1 null genotype, and C would be non-carriers of the genotype. O would be risk of lung cancer. So it seems as though everybody was on the right track, which is looking good. And then I'm not sure whether they would like a challenge of maybe formulating the research question.
Do you want them to have it in the chat as well?
Yes, if they're brave enough, that would be great. Otherwise, I can go through the review question.
Okay, I will let you know if there's anything. Thank you.
Sure.
Kimona, I don't see anything posted, so
Sure.
I
So
think you can
I'll
continue
take
for
it.
the sake of time.
Sure. So I'll go through the review question. So the review question would read, the population, since it's smokers aged 40 to 65, you can start with that. In smokers aged 40 to 65 years old, does the GSTM1 null genotype modify the association between smoking and lung cancer risk? So this is a gene environment interaction, and that's why we use the term modify. So another type of huge review question you could ask yourself is a pharmacogenetic related question. So how the genetic variation affects response to drugs or treatment in terms of the drug bioavailability. And here I'll use an example using the PICO mnemonic, where P is the population, which are patients with uncomplicated malaria treated with the drug, and then E, the ABCB1 transporter gene polymorphism, C would be the wild type genotype, and the outcome here would be two types of outcomes. So the day 7 plasma concentration, as well as the malaria treatment outcome. Is there a recurrence or is there a cure? And using these PICO elements, or PECO elements rather, when we formulate the question, we would ask, what is the effect of the ABCB1 gene polymorphism on day 7 plasma concentration and malaria treatment outcome? So I'm going to explain to you how to conduct a huge review. So initially you would start with the literature search, and you would use, as mentioned before, an extensive search, including multiple databases, such as PubMed, Huge Review Navigator, etc. You would combine the disease and the gene terms. You would also use mesh terms, perhaps, for the diseases. And include non-English articles. The reason why you would include non-English articles would be because there's a lot of genetic studies that are published in, for example, China. And so those articles aren't in English. So to be very thorough and extensive, you would include that in your review. You would also go on to include gray literature, and this would include the conference proceedings. In most cases, genetic studies don't often get published, so this is where you would find this information. And lastly, you would use citation tracking or snowballing, and this is to find missing or missed data or missed studies, yeah. And once you've done your literature search, you would then go on to do the data extraction, and this would be done by 2 independent reviewers using a standardized data extraction form, and it would include details such as the genotypes, the outcomes, confounders such as age or ethnicity, as well as sample sizes. So this is very important, addressing and assessing the potential biases. So you would do this to check the study quality as well as the methodology. And so the risk of bias tools or critical appraisal tools will summarize the study validity and address multiple testing. It would also look at various biases, such as selection bias, whether it affects the representativeness of the sample, information bias, were there in fact errors in data collection, so genotype misclassification. It would look at the confounding bias, such as population stratification. So you'd look at ethnicity, age. And lastly, you would also check the Hardy-Weinberg equilibrium. And on the right here, this is a very nice paper to go through if you're wanting to look for critical appraisal tools on human genetic studies. So the Hardy-Weinberg equilibrium states that a population's allele and genotype frequencies are constant unless there is some other type of evolutionary force acting upon them. So you would want a normal population of the controls. So here you have two types of equations, which is a allele frequency equation, and then the second equation is your genotype frequency, and this is what you would use to calculate the Hardy-Weinberg equilibrium. So there are 5 assumptions that must be kept in order for the Hardy-Weinberg equilibrium to happen. And this is the fact that no selection of the studies would have been done. There must not be any mutations or migration. You would need to include a larger population because small populations are vulnerable to the genetic drift and random mating. And the reason why you would use a Hardy-Weinberg equilibrium is to detect whether there's any bias between the control groups. So in this case, is there selection bias of the types of controls that you included or population stratification bias? You would also check the genotypes. So the control group genotype distributions of using the Hardy-Weinberg equilibrium. And if there is a deviation of the Hardy-Weinberg equilibrium, then you would conduct sensitivity analysis, whereby you would exclude a particular study if it doesn't adhere to the Hardy-Weinberg equilibrium, conduct the meta-analyses and see if you get the similar result with and without the study. So this brings me to the meta-analysis component. Just to mention that the effect size for meta-analysis of huge reviews is usually an odds ratio. And you would then, once you decide on what effect size you are going to be using, you would need to decide on the type of model, whether it's a fixed or random effects model. So a fixed model would assume that all studies have the same underlying mechanism, and a random model would show that there's variations in the PECO elements, as mentioned before, and that can be as a result of the weighting of the studies. You then go on to assess the heterogeneity of the studies, and you can use this, the I-squared test and the Cochrane Q statistic. So the heterogeneity is just to check where there's any differences between the studies, which is really important when conducting a meta-analysis. Just to note that small studies should be approached with caution due to the risk of spurious findings. And it is important to conduct a sensitivity or subgroup analysis, or if in fact you're able to, a meta-regression analysis. And this is to assess the impact of the studies in terms of the susceptibility to bias. And then lastly, you have to ensure transparency in the statistical choices that you have made to include into the review. So the results of the meta-analysis are presented on a forest plot, and it is called this because the structure resembles a forest. Each row represents an individual study in the analysis. So here you have 9 studies, as with like a branch on a tree, contributing to an overall structure or body of evidence. So the forest plot depicts the effect measure. In this case, it's the odds ratio, as indicated here, and as well as the 95% confidence intervals, which is indicated as the black lines here. So just to also mention, within that, there's these little blocks or squares, and those represent the individual study effect, which is the size of the block varies to reflect the weight at a particular study has that in the overall analysis. And just to mention, the smaller blocks, which have less weight, generally will have a larger confidence interval than the larger blocks. And the most important thing to note here is the diamond, which is the overall effect measure. So the outer edges of the diamond represents the confidence intervals. In this case, they used a random effects model. So there must have been some differences or variations in the study. And they were looking at the association of a polymorphism with lung cancer risk. And when you look at the diamond, which represents the overall effect, you can see it's got a 2.07 increased odds of association. The polymorphism has a 2.07 increased odds of association with lung cancer risk. And this is statistically significant because it does not include one. If it did, in fact, include one, so how you would know this is looking at the confidence intervals. The lower confidence interval in this case is 1.32, so it does not include one, and therefore it is significant. So this is just to give you an understanding. Later on, we'll go into the details using examples of actual huge reviews. Oh, and I forgot to mention, this is very important, the line of no effect. So there's a site on the CDC website called the Huge Review Navigator. The link is at the bottom here if you're interested. So this navigator uses a web-based application that encourages the exploration of the literature on human genetic association studies using a database that's continuously updated from PubMed. So it's free and it's user-friendly. So you can go and look here at the available huge review literature. You can also look at the diseases and the genes, which I'll show you now. So if you're looking at a particular disease, you go to Phenopedia, you type in your disease of interest. In this case, I used diabetes as an example. And it gives you a list of the diseases. So there's different clinical representations of diabetes. My interest was in diabetes type 2, so I selected that. And it shows you the list of associated genes on the far left. And here you can see that there's 140 genes that have been reported with diabetes mellitus type 2. And what's great is on the right of it, the next column shows the total publications. So those are the publications related to those particular genes. And if there is in fact a meta-analysis that was done, this would also be listed here. I do want to make mention that the site has been discontinued. However, you are still able to search for the data until 2029. So it's a great resource if you're just starting out a study and you want to know what kind of genes are associated with a particular disease. You could also go from the gene perspective. So you put in your gene of interest And in this case, you want to know, so I picked TNF gene, and then there's various variants of it, and I looked at the CD40 option. And here you can see the types of disease associated with that particular gene. So it's 110 diseases associated with that particular gene. And as mentioned before, the total number of publications and meta-analysis if it was conducted. So it's a great tool to use, especially if you're starting out a huge review or a genetic study and you want to know more about the disease or the gene of interest. So I highly recommend using it. Oh, and so what's great is if you click on the publications, it takes you directly to the PubMed article. So you can read up on it, download it, and so on. So it's very user-friendly. So to make this concrete, let's look at some published huge reviews. The important thing is to see how the methods we've just discussed play out in real reviews, from framing the question to pooling the evidence and drawing meaningful contributions. So in this example here, we're looking at a huge review of a polymorphism and non-alcoholic fatty liver disease. So they looked at different types of diseases, three to mention. So it's non-alcoholic fatty liver disease, NAFLD, the simple steatosis, and non-alcoholic steatohepatitis. And here you can see the Prisma flow diagram, which shows you the databases that they searched, the types of articles they excluded, and the reasons as to why they excluded these articles. And at the bottom of the flowchart, you can see that they included 23 trials in the meta-analysis. However, for each disease, there was a different number of publications. So for NAFLD, there was 23 publications related to that polymorphism. So table 1, traditionally, you would use your table 1 for your data extraction form. And so this would be the characteristics of the study. So the type of data you would extract would be the country or the region, the ethnicity, because this is genetic study, so you want to see where there's differences in ethnicity, the source of the control, whether it was population-based or hospital-based, as well as the genotyping method. It also goes on to tell you the number of cases and controls. And here on the far right, you can see the quality score, which they indicated. So I think they used their own in-house risk of bias tool. It wasn't a Newcastle Ottawa scale. So in this case, I think their risk of bias was of good quality. Okay, so there we go. So in the second table, you can see the distribution of the alleles and genotypes of the polymorphism in the NAFLD studies. So it details the number of cases and controls, the genotype in the cases, as well as the genotype in the controls, as well as the alleles, the G allele and the C alleles. So you would use this information when you are calculating the Hardy-Weinberg equilibrium. And so what they would use is a chi-square test using the Hardy-Weinberg equilibrium information. And here you can tell by the p-values whether it's significant or not. So a p-value of less than 0.05 would indicate that there's a significant, and that would tell you whether the Hardy-Weinberg equilibrium whether there's a deviation in the Hardy-Weinberg equilibrium of your controls. So in this forest plot, it shows that there's a significant association. If you look at the diamond, the overall effect, and you can see that it's an odds ratio, there's a significant association between the NAFLD, which is the non-alcoholic fatty acid liver disease risk, and the polymorphism. So everything is to the right of the forest plot, indicating, and you can see based on the odds ratio, it was a 3.41 odds ratio, not including one, so therefore statistically significant. And all the studies seem to have overlapped, so that's great, meaning that there's not a lot of heterogeneity among the studies. The second forest plot indicates the polymorphism was also significantly associated with the NASH. So the non-alcoholic, the NASH disease, and so it was. You can see that it was also significant in all of the genetic models. So when looking at the simple steatosis risk, you notice that the polymorphism was only significantly associated with the risk of simple steatosis in the allele contrast, and it had no effect in the other genetic models. So this is another example, and this was a huge review looking at polymorphism, the GST polymorphism, and prostate cancer. The image on the right is the Prisma flow diagram, and apologies for the poor quality, but here it just indicates that they included 37 articles in this review. So table 1 shows the frequency of the polymorphisms, genotypes, and the major ethnic groups. So in this case, it was Caucasian, Asian, and African American. And you can see that they have the GSTM1 genotype, the GSTT1 genotype, as well as the GSTP1 genotypes. And in this forest plot, you can see that the overall effect was to the right of the line of no effect. So this would mean an increased odds ratio. And so as indicated, it shows that there's a 1.33 increased odds of association between the GSTM1 polymorphism and the development of prostate cancer on a wide population basis, because it includes both Caucasian, all of these racial descents. So in the GSTT1 and the GSTP1 polymorphisms, you can clearly see that the overall effect falls on the line of no effect for both of these polymorphisms. And this indicates that there's no association between these two polymorphisms and prostate cancer risk. In the third and last example, we're going to look at the polymorphism RS108653331 in 2P15 and ankylosing spondylitis. Ankylosing spondylitis is a chronic inflammatory disease, and that primarily affects the spine, causing chronic back pain and stiffness. So this review was the most recently published review. I think it was published in 2020, as opposed to the other reviews that I mentioned. So you can see that the abstract looks very much different to the other reviews. And so this review investigated the correlation between the polymorphism and the risk of ankylosing spondylitis. It also details quite nicely the databases that they used for the methods and the screening process and the effect measure. So in this case, they're looking at the odds ratio, and it tells you that they did conduct a sensitivity and subgroup analysis, which is great. And they also looked at the publication bias. So as mentioned before, the Prisma flow diagram shows that the numbers of articles included and the reasons for exclusion. So here, table 1 is the characteristics of the included studies. They looked at the gender, age, and you can see that they mainly included case control studies, and they detailed the genetic distributions of the genotypes, which is really important, as mentioned before, for determining the Hardy-Weinberg equilibrium of the control group. And as mentioned before, a p-value of less than 0.05 indicates a significant difference. And we don't see any, I think there was one study, Lynn, that had a significant difference. So table 2 is the results of the polymorphism and ankylosing spondylitis susceptibility. And here you can see that they looked at the effect measure, the odds ratio, for the recessive, dominant, as well as the alleles. And so there were 9 studies that looked at that. And then when you look at the pooled analysis, this was an odds ratio that was significant because it included did not include 1. And so it was an odds ratio of 1.295. You can also see that the heterogeneity was a bit high. It was a moderate to high heterogeneity of 73.70. And they even looked at the publication bias, as mentioned before, and there was some publication bias seen. So if there is publication bias and a high heterogeneity, you would go on to do sensitivity analysis, where you would exclude one study at a time and redo the meta-analysis to see whether there's a change of the effect direction. So this forest plot shows an overall odds ratio of 1.29 with a confidence interval that does not include 1. Therefore, it is in fact significant. And so the heterogeneity, as mentioned before, was 73.7. So that's an indication that it's on the higher side, with a p-value that indicates that it's quite significant. This could be as a result of a study by Zhang A. And so, as mentioned before, you would go on to exclude this study and see whether the effect measure direction changes. So I think in this case it didn't, and so that's why they left the study in. So overall, you can see that there was a 1.29-fold increase in association between the polymorphism and the development of ankylosing spondylitis. And that brings me to the end of this webinar. If there are any questions, please feel free to ask. Thank you.
Thank you very much, Kimona, for that informative session. I don't know if it's the weather, but I feel 1 degree smarter. So now over and above to the colleagues who have some questions or want to participate in like a discussion. If you want to say anything, please raise your hand or type it in the chat box. You can unmute yourself, Kim.
Okay.
Hi, good afternoon. Police, thanks, Kimona, for very interesting presentation, interesting topic. Yes, and it's the first time I attend the webinar about genetics reviews. Very interesting. May I have two questions, please? My first question is seeing the advice is to include the non-English publications. So I'm just wondering because I'm not come from the genetic fields. Doesn't a gene also have a different name in different language? So if we reviews and then in different language and how accurate in terms of translations. And can I also pose my second question?
Yes.
Okay. My second question is how is the certainty of evidence accessed in the huge reviews? Thank you.
Thank you. Thank you, Kim, so much for your questions. The first question you made mention of the gene names could differ from different languages, other languages, and that is actually a good point. So it will be useful to go and check whether the names, the gene names are different in different languages. So yeah, so thank you for bringing that up, Kim. So that's a good point that I didn't mention. The second question was regarding the tools, the critical appraisal tools. So there is the Newcastle Ottawa scale for regarding risk of bias that most reviews, huge reviews use. But when it comes to grading of these studies, it's not yet part of the huge review, and it's used. So that would answer the certainty in terms of the evidence. So it hasn't been done yet, and I think it leaves a space for that to be done and to be included in huge reviews. So at the moment in time, it's just risk of bias and not grade.
Thank you.
Yeah, thank you for your questions, Kim. Are there any other questions, Benny, in the chat?
No, there's nothing else. Thank you, Kimona. We appreciate you. Thank you very much for this informative session. If anyone wants to ask Kimona more questions, they can reach out to her. I will put the email address in the chat.